Viruses, viroids, and prions are infectious particles that are important in medical microbiology. Viruses are obligate intracellular parasites. They must live inside a host cell. They are submicroscopic. They're measured in nanometers and are only visualized using an electron microscope. They're pretty simple. They're composed of a protein coat, and that protein coat is called a capsid. The capsid gives each virus a unique shape. The capsid is made up of repeating subunits called capsomeres. The other part of a virus is the nucleic acid. Viruses have either DNA or RNA, but never both. And the DNA and the RNA can be in all kinds of forms. You can have double-stranded DNA, double-stranded RNA, single-stranded DNA, single-stranded RNA, linear DNA, circular DNA, linear RNA, circular RNA. So there's a wide variety of forms of the nucleic acid. The capsid plus the nucleic acid is referred to as a virion, or sometimes it's called the nucleocapsid. Some viruses have an envelope. Now, the envelope is an outer membrane that came from the last cell that virus infected. Viruses that have this outer membrane are said to be enveloped viruses. Viruses that do not have this outer membrane are non-enveloped or naked. Here is a polyhedral virus, and you see all of these little capsomeres that come together to make up the entire capsid. And inside this is a double-stranded DNA molecule. That would be a virion or a nucleocapsid. This would be a naked virus because there is no outer envelope. Viruses are very host-specific. They have an affinity for the surface of some host cell. So there are viral proteins or glycoproteins on the surface of the virus that recognize receptors on the surface of the host cell. Even within a host, viruses may be cell-specific, so only certain cells within the host may be infected by the virus. Bacteriophages or phages are viruses that infect bacteria. Viruses infect virtually every organism on the planet, even other viruses. Viral shape is one of the things that's used to classify viruses, and there are three basic shapes: helical, polyhedral, and complex. This is based on the capsomere arrangement. In helical viruses, the capsomeres are arranged in a spiral, so you end up with a rod-shaped capsid. Polyhedral viruses look like a geodesic dome. They are triangles that are fit together. The most common shape is the icosahedron, which has 20 triangles. And then complex viruses have several different capsomere shapes. So when you put these viruses together, they're neither helical nor polyhedral. A lot of these look like lunar landing modules. All viruses lack a cell membrane, but some viruses retain the cell membrane of their last host cell. This is the viral envelope. Viruses place unique glycoproteins in the infected cell membrane. These are these protein spikes. And when the virus leaves the cell, it takes this membrane with the protein spikes with it. The protein spikes are part of the recognition system for the next host cell. Envelope viruses also have matrix proteins between the membrane and the capsid to help hold the envelope in place. In 1966, the International Committee on Taxonomy of Viruses was developed. This committee was designed to put together a logical classification system for viruses. Since viruses are non-living, they do not fit into the Linnaean system of taxonomy. They looked at the type of nucleic acid in the virus, the presence of an envelope, the type of nucleic acid, is it double-stranded or single-stranded, linear, circular, and the shape and size of the virus. They came up with 22 viral families. They don't have viruses in kingdoms or phyla. Families was the best they could do in terms of organizing them. The DNA viruses include these families: the pox viridae, which houses the virus that causes smallpox. Herpes viridae contains viruses that cause herpes, both cold sores and genital herpes, chickenpox, mononucleosis, cytomegalovirus, and roseola. The papillomaviruses are the ones that cause warts. The polyomaviridae are very rare. They cause a disease called progressive multifocal leukoencephalopathy. This is a disease where white blood cells develop clusters in the brain, and it's always fatal. The Arenoviridae have a group of viruses that are involved in causing conjunctivitis and respiratory infections. And the Parvoviridae has a virus that causes erythema infectiosum. This is a disease where the individual's cheeks turn bright red. It's sometimes called slapped cheeks disease. This is kind of a wimpy virus. It only develops secondarily to some other viral infection. There are many more families of RNA viruses. The picoRNA viridae are very small viruses. That's what pico means. And then RNA tells you that they're RNA-containing viruses. Polio, hepatitis A, and viruses that cause the common cold are found in this family. The Calicifieridae has the norovirus in it. And the norovirus has spoiled many cruises for many people. This causes gastroenteritis. The Astroviridae also holds some viruses that cause gastroenteritis. Hepaviridae has the virus that causes hepatitis E. Hepatitis E is only seen in third world countries currently. The Togaviridae have viruses that cause various forms of encephalitis and rubella. The Coronaviridae have viruses that cause the common cold. Flaviviridae has the viruses that causes yellow fever and hepatitis C. And Retroviridae has the virus that causes AIDS. Orthomyxoviridae, also RNA viruses, has the virus that causes influenza. The Paramyxoviridae contain viruses that cause the common cold, measles, and mumps. Rhabdoviridae houses the virus that causes rabies. Bunyaviridae houses the viruses that cause California encephalitis and house the hantavirus. The Filoviridae, this is where we find the virus that causes Ebola, and also the virus that causes Marburg disease, which is a milder form of Ebola. Arenoviridae houses a virus that causes Lassa fever. Lassa fever is only seen in one particular location, Africa, the Lassa Valley. This is a hemorrhagic fever, so it's sort of like Ebola. And Reoviridae have viruses that cause encephalitis. Also, the rotavirus is here, and rotavirus causes diarrhea, particularly in young animals, including children. Viruses cannot reproduce themselves because they are not living. They have to be inside a host cell in order to do this. Basically, what they do is once the virus enters the host cell, it hijacks the host cell's organelles and metabolism and destroys the host cell DNA. This means that the DNA or RNA from the virus is the only controlling molecule in the cell, so the cell is forced to make new virus particles. The new virus particles then leave the cell, typically killing the cell. Bacteriophages have been the best studied, and so we have a good view of how they reproduce. And the lytic replication means that at the end, the cell is going to be lysed. There are basically 5 stages to viral replication. We start with attachment. The virus and the host cell must recognize each other. Then the viral nucleic acid has to enter the cell. In the case of bacteriophages, this is DNA. There are enzymes with the virus that then destroy the cellular DNA, leaving the viral DNA as the only coding agent. The viral DNA causes the host cell to replicate the nucleic acid and the capsomeres for the virus. This is the synthesis stage. In the assembly stage, the phage components are put together into new virus particles, and then the virus is released. The virus leaves the cell, and because this is a lytic phage, it's going to leave the cell by lysing the cell. You can see that the bacteriophage is a complex virus. It's going to attach to the cell surface, and it injects its DNA into the host cell. So now we have the bacterial circular chromosome and this DNA, which is from the virus. Notice the first thing that happens is the bacterial DNA becomes destroyed, leaving this as the only DNA to control cell activity. Synthesis occurs as we make more DNA and we make all the various capsomere pieces. Then during assembly, all of the capsomere pieces are put together and the DNA is packaged back up and you make a mature virion. At this stage, the cell lyses and these virus particles are released, and each virus particle can now go and infect another bacterial cell. Lysogeny occurs when the virus infects the cell but does not reproduce. The lysogenic replication cycle is what ensues. These are called temperate phages because they live temperately inside the bacterial cell and don't destroy it immediately. In the case of a temperate phage, viral DNA enters the cell, but the cell DNA is not destroyed. The viral DNA becomes what's called a prophage. The viral DNA remains inactive because some of the genes code for a repressor protein that prevents the bacterial DNA from being destroyed. This DNA from the virus actually inserts on the host cell, and as the cell replicates, all new daughter cells will have the prophage as part of the circular chromosome of the bacteria. At some point in time, a temperate phage can become a lytic phage. This is called lysogenic conversion or induction. Things like ultraviolet radiation or chemicals may act as the induction agent. At that point, the prophage comes out of the host DNA and the virus completes the lytic cycle. So here we have the lambda phage, again another complex phage. It can be lytic. It can put its DNA in. The DNA can destroy the host cell DNA, make new virus particles, and the virus particles are released from the cell. Or it can be temperate. That is, instead of destroying the DNA of the host cell, the prophage inserts on the chromosome. This means that as these cells reproduce, every new cell has the bacterial DNA plus the prophage DNA. All right, and this then leads to a population of cells that all have this prophage. The prophage may then break out at some point in time as a result of induction, and it will go back into the lytic cycle. Now, sometimes the prophage brings with it genes that give the bacterial cell some special properties. Some prophages are necessary for certain bacteria to become more pathogenic because it allows them to produce certain toxins. Animal virus replication is very similar to bacteriophages. There is the attachment cycle where receptors on the cell recognize capsid proteins or envelope spikes on the virus. This allows the virus to attach to the cell. Entry and uncoding is a little different. Some naked viruses may simply directly penetrate the cell, sending in their DNA or RNA. Some viruses are taken in by endocytosis. Envelope viruses may be taken in by membrane fusion. If the virus is taken in whole, at some point the capsid has to be removed so that the viral nucleic acid can enter the cell. And this process is not clear as to how this happens. Somehow the capsid is digested off of the virus. So here you see a direct input. The viral genome is simply inserted into the host cell. Or here is the membrane fusion where you have an envelope virus. And again, the entire virus goes in, so at some point we have to uncode it to get the viral genome out. Or here is a case of endocytosis, where the virus is taken in by endocytosis. Then the virus particle is released inside the cell. And again, this magical uncoding has to occur. Animal viruses replicate differently because they have different nucleic acids in them. The DNA viruses may have either double-stranded DNA or single-stranded DNA, and both of these have different mechanisms for taking over the host cell. But no matter what, the DNA migrates to the nucleus and controls the cell's activities from there. With RNA viruses, we also have double-stranded RNA and single-stranded RNA. And the single-stranded RNA can be either positive sense RNA or negative sense RNA. If the RNA is single-stranded and positive sense, a ribosome can immediately attach to that RNA and begin protein synthesis. If the single strand of RNA is negative sense, the cell first has to make a complementary copy of the RNA, and that is the piece of RNA that the ribosome can attach to. Again, in either case, the control of protein synthesis occurs in the cytoplasm. Retroviruses are a little different. They're RNA viruses, but they carry with them an enzyme called reverse transcriptase. Now, transcription is what occurs in the nucleus when we take DNA and make messenger RNA. So reverse transcriptase is the reverse of that. We take the viral RNA and we make DNA against it. From this point on, these retroviruses act like DNA viruses. That DNA migrates to the nucleus and controls the cell from there. DNA viruses will assemble in the nucleus and RNA viruses will assemble in the cytoplasm. If we have an envelope virus, it will have placed protein spikes in the cell membrane and it will bud through the membrane. If it's a naked virus, it may leave the cell through exocytosis, just be ejected from the cell without a membrane, or it may directly lyse the cell so the virus particles are released. In any case, cell death is the typical end of a viral infection. For one thing, the nutrients of the cell have been depleted by the viral replication. The entire cellular metabolism has been aimed at making new virus particles. Also, if the virus buds out, taking membrane with it, then we've got a loss of membrane, and pretty soon the cell is too small to exchange a sufficient number of nutrients across the membrane. Of course, if the cell lyses to release the viral particles, then the cell is dead. Now here's an example of an enveloped virus being released. This is an RNA virus. The capsomeres are placed here against the cell membrane with those protein spikes, and as we package up the virus, we simply wrap the capsomere around it and we take the envelope with us. And here is a photomicrograph of that occurring. If we look at the infection of a cell with an envelope virus, here are the protein spikes attached to the receptors on the cell so that the virus is taken in. This would be by endocytosis. The vesicular membrane degenerates, the membrane around the virus degenerates, and the capsid somehow degenerates, releasing in this case RNA into the cytoplasm of the cell. This RNA generates more RNA molecules. It is used by the ribosomes to make the capsid pieces, and it is used by the ribosomes to make the new protein spikes. The new protein spikes are inserted on the cell membrane. The capsid is put together around the RNA, and as the virus goes up to the cell, it simply buds out of the cell, so we have a new virus that is now enveloped. Animal viruses can display something called latency. We see this in chickenpox and herpes, for example. In this case, viruses enter the cell and may cause disease, but stay behind in the cell and become dormant. This is called a latent virus or a provirus. Latent viruses do not necessarily incorporate their DNA into the host cell like a lysogenic virus. However, if the provirus does incorporate into the host cell DNA, it's a permanent incorporation. There is nothing that will induce the viral DNA to leave the host cell DNA. Viruses can remain dormant for years, but can become active at any time. These are called persistent infections. If any of you have herpes simplex and have fever blisters, you know that they come and go, and that is an example of a latent virus, and you have a persistent infection. In normal cells, the control of cell division is very tightly controlled. There are proto-oncogenes that control cell division. And when you're a small multicellular organism developing, these proto-oncogenes are very active because you need lots and lots of rapid cell division to grow. But as cells differentiate, as they specialize, these proto-oncogenes become repressed so that cell division becomes more controlled. If these proto-oncogenes are reactivated, they become oncogenes. This is when you get unrepressed cell division. Neoplasia is the condition of uncontrolled cell division, and this can lead to tumors. Benign tumors stay encapsulated, stay in one place, and other than sometimes causing pressure, really don't cause much damage to the body. Malignant tumors, however, invade surrounding tissue, and because the cells don't stick to each other well, they can get in the blood or lymphatic system and travel to other organs in the body. We call this metastasis. Now, about 20 to 25% of human cancers are virus-related. There are a lot of reasons they think viruses can cause cancer. DNA viruses may carry with them oncogenes that will then cause unrepressed cell division in the infected cell. Viruses may promote oncogenes that are already in the cell. Or if they become proviruses, they can insert on the host cell DNA and they just randomly insert. If they insert in the middle of one of these repressor genes, then the repressor gene becomes inactive and an oncogene is now activated. Viruses are challenging to culture because they must grow in living cells. Bacteria can act as hosts for bacteriophages. We can also use live plant and animal cells. However, it becomes a little unethical if we're trying to grow human viruses. It turns out that embryonated chicken eggs are a really good source of tissues for viruses to grow on. The developing chicken has lots of different tissues, and the cells are undifferentiated cells, so they have a wide host of receptors on them. So lots of different viruses can be hosted by an embryonic chicken. The other way to culture viruses is using cell cultures, and there are basically two kinds of cell cultures. The diploid cell culture comes from embryonic plant and animal cells. These cells will go through about 100 cell divisions before they die, so these cell cultures are limited in how long they will last. Continuous cell cultures are cell cultures that develop from tumor cells. The oldest and best known is the HeLa cell line. This is named for Henrietta Lacks. She had cervical cancer. She died in 1951, but her cervical cancer cells were harvested, and we still have the HeLa cell line as a line for growing viruses. When you are identifying viruses, one of the things you can do is look for changes in the infected cell. This is called cytopathic effect. Since DNA viruses assemble in the nucleus, looking for changes in the nucleus is one way you can tell it's a DNA virus. If you see changes in the cytoplasm, that's an RNA virus. Also, the type of tissue they infect tells you something about the virus. Viroids are small circular pieces of RNA that are infectious and pathogenic for plants. They cause things like plant wilt or they stunt growth in plants. They affect our food supply, but so far they have not infected humans. Viroid-like agents are other forms of infectious pathogenic RNA particles that affect fungus. Again, so far we have not seen any crossover into humans. Prions are proteinaceous infectious agents. They were first described in 1982. The concept of a prion was resisted by scientists for many years because how could a protein make more protein? We were used to a model where nucleic acids were responsible for protein synthesis. It turns out that all mammals make a cytoplasmic protein called PRP. Now, PRP is anchored in a lipid raft, and you'll remember that in eukaryotic cells, we could have clusters of lipids and proteins that were always together. They could float around in the phospholipid bilayer, but this little cluster always was consistently together. That's what a lipid raft was. It turns out that PRP plays a role in normal brain activity. We don't exactly know how it works, we just know that it plays this role. Now, what a prion does is it encourages PRP to fold differently than normal. Normally folded PRP will be just fine and work just fine in the brain, but if a prion comes in, it's sort of like a bad seed. It tells the normal PRP to fold differently and become this abnormal PRP. This improperly folded PRP clumps together, causing neurons to stop working, and when neurons stop working, they die, so this leaves holes in the brain. This causes a condition called a spongiform encephalopathy. Under normal conditions, the other molecules in the lipid raft help keep PRP properly folded. However, some people carry a mutation for PRP. This means that their PRP could be folded differently, but these other molecules keep it in line. This means that about 40% of humans have a mutation and are susceptible to prions. There are several prion-associated diseases. Bovine spongiform encephalopathy is also known as mad cow disease. Scrapie is a spongiform encephalopathy seen in sheep. Kuru was a spongiform encephalopathy seen in Polynesian natives that ate their dead. Since this practice has been discontinued, Kuru is no longer seen. Chronic wasting disease is a spongiform encephalopathy seen in deer and elk. And then variant Creutzfeldt-Jakob disease was something that was seen because, while we didn't think it could happen, prions seemed to have jumped species. Scrapie infected sheep were used to make cow feed, which developed mad cows, which were used to make hamburgers and other meat products, which were eaten by people. And this is where we see the variant Creutzfeldt-Jakob disease. Prions are not destroyed by normal cooking or normal sterilization methods. If you incinerate them, that is, if you burn them completely, that seems to get rid of them. And they've now discovered that autoclaving them in concentrated sodium hydroxide destroys them. There is no known treatment for prion diseases, and variant Creutzfeldt-Jakob disease is very rapid in its progression. Most others are much slower in their progression. Because scientists are now looking more closely at prions, they're wondering if they may not be involved in certain other neurological disorders like Alzheimer's or Lou Gehrig disease. It's yet to be seen.